Hello, today I'm going to tell you a story about the effect of different lengths of CAG repeats, 16 and 128, and the first exon of the Huntington gene, when integrated into the Drosophila melanogaster genome. In humans, over 39 CAG repeats in this area cause Huntington's disease, a devastating neurodegenerative disease that causes, among other things, uncontrollable jerky movements. These CAG repeats get translated into polyglutamine repeats in the Huntington protein and cause the protein to aggregate with neighboring proteins at the region where the polyQ repeat occurs. This aggregation disrupts cell functionings, such as signaling pathways, as shown in Figure 1, which is taken from Lee et al., 2004, which in turn causes neurologic symptoms. Drosophila are an ideal model organism to study this disease because they have a short generation time, well-studied genome that can accommodate transgenes, and a nervous system similar to that of humans that allows them to express the movement-related behavioral deficits that characterizes Huntington's disease. For my experiment, that I performed with Rachel Gerber and with the guidance of Dr. Kaczynski Collins, I induced the expression of 16 polyQ repeats in one line of flies and 128 polyQ repeats in another line of flies, with the hope of studying behavioral deficits between them. These number of repeats were chosen because the former is well below the threshold for Huntington's, while the latter is well above this threshold. In order to introduce this gene into the Drosophila genome, the GAL4-UAS system with an actin driver was utilized. With this system, the transgene is integrated into all cells, but only expressed in the cells that contain a tissue-specific driver, in this case actin. However, since actin is expressed in all cells, so is the polyQ mutation. Figure 3 contains an image captured by Charles Mazel of a Drosophila larva where actin was tagged with a fluorescent marker, proving the ubiquity of the protein. Female flies with the genotype UAS-Q16/CYO and UAS-Q128/UAS-Q128 were crossed in duplicate with males with the genotype actin-Gal4/CYO. The progeny expressing the transgene could easily be identified from both crosses because they had straight wings, as compared to their curly-winged, non-expressing siblings. 7 days after the cross was started, I counted an average of 31 flies expressing the Q16 genotype and 0 flies expressing the Q128 genotype. This led me to conclude that the Q128 mutation is deadly when expressed in all cells. In the future, I would like to investigate at what stage in development the Q128 mutation killed the Drosophila. To do this, I would repeat the same cross I did in this experiment, but tag the polyQ repeat with a fluorescent marker such as GFP and image the eggs, larvae, and pupae that result to see if they contain the Q128 mutation. After performing that experiment, I would go on to conduct a qPCR experiment to determine the expression levels of the altered Huntington gene in the latest stage of development of surviving Q128 Drosophila versus Q16 Drosophila to determine if differences in expression levels are contributing to the lethality in the Q128 flies. Lastly, I would again like to repeat this cross, but instead of using an actin driver, I would use an LAV driver, which induces expression only in neurons. Hopefully, this will stop the mutation from being lethal and allow for movement behavior to be assessed using a negative geotaxis assay.